Thank you for joining me today. 2021 has started out to be a very exciting time here at Dyadic. Earlier this month, during the ZAPI Stakeholder Conference, the C1 technology was featured for its robustness, its versatility, its productivity, its stability, its efficacy, and its safety in the animal studies that were carried on by ZAPI over the last several years. Our technology platform seems to be advancing in the animal health industry, in human health industry, and infectious disease. Certain statements contained in this presentation are forward-looking statements. We have a solid financial position. We're able to create value through technology licensing, co-development partnering, and wholly owned product development. We supplement that with fully funded research and development with our collaborators from big pharma, biotech, and governmental agencies on a global basis. The C1 platform has demonstrated the versatility to efficiently produce large quantities of more affordable vaccines and drugs for ourselves and for our collaborating partners. A growing number of scientists globally are recognizing that C1 is ideally suited to help meet the global demand for multivalent vaccines and multi-antibody cocktails, as well as other therapeutic drugs. They recognize that we have developed an efficient biomanufacturing platform to serve a global population in larger quantities, more affordably, at flexible commercial scales. They also recognize the speed at which we can create C1 cells that are stable, highly productive, and highly scalable. We have partnered with some of the leading collaborators in both animal and human health and with governmental agencies. And we see that the 2021 is going to be a defining moment for C1, Dyadic, humankind, and our investors. We have and continue to develop strong relationships with top-tier global partners in animal health, human health, and infectious disease. We've established global partnerships in Asia, in US, in Europe, and in other countries. We have a highly experienced and energized professional management team and a world-class board of directors and advisors. Together, we are applying C1 in many different directions, but we're focused on one thing: advancing science, creating business opportunities, creating value for Dyadic shareholders, and making healthcare more accessible and affordable to patients on a global basis. The addressable opportunities of where we can apply the C1 technology are enormous. We're applying C1 in animal health, human health, infectious disease, and other markets. Where we apply the technology, we either apply it for ourselves to develop our own product opportunities, or we can apply it in collaboration with big pharma, with biotech, and governmental agencies. In all cases, we're trying to create products that can be made in larger amounts at lower cost at flexible commercial scales. In addition to large volume, low cost is enablement. And there are many opportunities that we're being confronted with, or opportunities that are being brought to us for enablement, where other technologies have failed to be able to produce sufficient quantities to allow product development to move forward, let alone commercialization. And that's one of the key areas where we're focused on. Our focus for 2021 is to build on the enormous momentum that we built in 2020. That momentum was based on stellar scientific data that came from the ZAPI program, the IIBR program, our own internal funding, our collaborations with big pharma and animal health companies, and with governmental agencies. We're advancing internal and external animal and human health towards preclinical trials, and we're moving C1 into its first cGMP manufacturing to produce clinical trial grade material for a potential phase 1 human clinical study with our own C1 RBD antigen against the COVID-19 pandemic. 2021 is a very exciting time here for us at Dyadic as we continue to prepare for our first potential human phase 1 clinical trial using a Dyadic-owned C1 cell manufactured SARS-CoV-2 receptor binding domain RBD recombinant antigen vaccine product candidate. This will demonstrate the safety and efficacy or proof of concept in humans. We're expanding the RBD antigen portfolio so that we can quickly respond against SARS-CoV-2 variants. We anticipate a potential phase 1 human clinical trial in the EU with a C1 cell manufactured SARS-CoV-2 monoclonal antibody. We continue to advance commercial recombinant antigen vaccine candidates into animal trials with global animal health companies. And we generate additional safety and efficacy data within ZAPI for the C1 produced SPV and RVFV recombinant antigen vaccines. On the human health side, we continue to advance immunotherapeutic monoclonal antibody with a top-tier biopharmaceutical company with registrational potential in oncology. We're continuing to advance immunotherapeutic bispecific monoclonal antibody with a top-tier biopharmaceutical company with a registration potential in autoimmune disease. And part of our glycoengineering effort, we're using nivolumab, Opdivo, as a potential biosimilar mAb product candidate. Our strategic collaboration with the Israel Institute for Biological Research, the IIBR, has helped to position Dyadic to enter human clinical trials. The IIBR demonstrated the C1 cell-produced SARS-CoV-2 receptor binding domain, RBD, can be manufactured in large quantities at low cost, which neutralizes the COVID-19 virus. The IIBR mice trial data demonstrated that the C1 cell produced SARS-CoV-2 RBD antigen, the C1 RBD, generated high levels of antibody binding and neutralization. Transgenic mice expressing human ACE2 were vaccinated with the C1 RBD, which were challenged with live SARS-CoV-2 virus. The IIBR transgenic mice expressing human ACE2 challenge data is expected in Q1. One of the many benefits that we derived from C1's excellent performance in the ZAPI program were building the relationships with scientists, with universities, and with biotech and pharmaceutical companies that were involved in that program. 3 of those scientists who saw the safety, the efficacy, and the productivity of C1 versus the other cell lines decided to join with us in the development of the Dyadic C1 RBD protein-based recombinant vaccine. We've now brought Parexel in to support us with regulatory and CMC consultation, CR2O for coordination of the potential clinical trial in humans, and BTG, which is a biotech pharmaceutical company, division of Ferring Company, that's a CGMP manufacturing of toxicology and clinical trial material. The infrastructure that we put in place through this relationship can be repeated time and time again. And we've already started to repeat that with the variants from the SARS-CoV-2 South Africa variant, UK variant, and South America variant. And we'll talk more about that coming up. I'm excited to share with you the strategy for the development of a tetravalent recombinant variant antigen SARS-CoV-2 vaccine product candidates. This would be up to 4 critical variant RBD antigens in a preparation produced from C1 cells called the Dyadic 4TVA antigen-encoded SARS-CoV-2 vaccine candidate. As you can see, we can rapidly engineer C1 cells to produce antigens at very high levels and at low cost, as demonstrated by the earlier program. In addition to the vaccine opportunity, we're working on monovalent and multivalent potential recombinant variant monoclonal antibody SARS-CoV-2 product candidates. We've recently executed a material transfer agreement with a top-tier pharmaceutical company who's going to perform a fully funded research and evaluation of a C1-produced SARS-CoV-2 monoclonal antibody. In addition, we anticipate entering into a fully funded collaboration with former ZAPI EU scientists and other parties to express one or more SARS-CoV-2 monoclonal antibodies with the objective of leading to a potential second phase 1 human clinical trial of a single mAb or a cocktail of more than one mAb expressed from C1 cells. As I mentioned earlier, the ZAPI Stakeholder Conference was held earlier this month. The data that was demonstrated and presented during that conference by the scientists that were involved in ZAPI, including Ronan Shellett, our CSO, and the 3 coronavirus experts that we spoke about earlier that are involved in the Dyadic vaccine programs, led to the fully funded collaborations with several leading global animal and human health companies and governmental agencies. ZAPI is continuing to fund additional animal studies in 2021 with the C1 produced SPV vaccine and the RVFV vaccine produced from C1. One of the data points that was presented within ZAPI was that C1 produced SPV was 300 times greater than the SPV produced from insect cells or baculovirus cells. Being involved in ZAPI has put us in a position and has played an important role in combating potential pandemics for today and future pandemics for tomorrow. We continue to advance and spend research and development dollars to advance the C1 platform and to transfer technology to potential producers of cGMP material for ourselves and potential collaborators. We've been working on glycoprotein engineering of C1 cells to advance Dyadic's nivolumab Opdivo biosimilar monoclonal antibody as a proof of concept for production of mAbs. The protease engineering, or the knockouts, or the reduction of protease activity, has been very successful. The development of the SARS-CoV-2 RBD and future RBDs of the different SARS-CoV-2 variants, influenza, and other infectious disease pathogens that we're exploring. We announced recently a fully funded program by Turtle Tree Scientific to produce growth factors from C1. And of course, we have a variety of fully funded proof of concept protein expression programs that we expect to lead to potential commercial opportunities, just like the ones mentioned earlier for oncology and rheumatoid arthritis. In 2020, we conducted tech transfers of C1 into a number of organizations, including CDMOs and pharmaceutical companies, including WuXi Biologics. In 2021, we've already completed the first engineering run and recently initiated the second engineering run with the objective to produce Dyadic SARS-CoV-2 recombinant antigen under cGMP in support of a planned phase 1 clinical trial in the development of additional monovalent and tetravalent recombinant variant antigens for SARS-CoV-2 vaccine product candidates. As noted earlier, the model protein that we're using in the glycoengineering program of C1 cells is nivolumab. Nivolumab is an anti-PD-1 IgG monoclonal antibody, potential biosimilar to Opdivo, which is an $8 billion drug marketed by Bristol Myers Squibb. If successful in making a C1 glycoengineered cell, they can mass produce with the right qualities and characteristics at high levels with good stability. We'll be able to make multiple new and/or biosimilars of potential monoclonal antibodies or bispecific antibodies or Fc fusions that need human-like glycan structures for function. Let's now look at some of the C1 cell high-value recombinant protein production platforms' competitive advantages. Let's now talk about some of the properties of what C1 cells can do. C1 is a filamentous fungus. It has very high levels of protein production. It was initially developed to produce large quantities of low-cost enzymes for textiles, biofuels, pulp and paper, etc., at enormous industrial scales, up to 500,000 liters. It's proven to be very low cost, high yield, high purity, and scalable, robust system with improved manufacturing and downstream benefits. C1 has multiple genetic tools associated with the cell line that can be used to program C1 cells with genes of interest to produce a variety of biologic products from vaccines, monoclonal antibodies, and other therapeutic proteins. As we mentioned before, we've manufactured SARS-CoV-2 antigen vaccine candidates that are now in late-stage preclinical development and heading towards toxicology, cGMP production for anticipated use in a phase 1 clinical trial. Industrially proven hyperproductive C1 cells offer competitive advantages over certain cell lines. Example on the downstream processing. In the case of E. coli produce proteins, you have to remove endotoxins. In the case of C1, there are no endotoxins to remove in the first place. So there's a savings in the cost and downstream, and the recoveries are higher. Same thing with CHO cells. They have viruses. C1 doesn't have viruses. Productivity, the yields in grams per liter per day are potentially far greater in a shorter amount of time. The media that you use to grow C1 is basically low-cost, chemically defined synthetic media, about 1/20th the cost of the media used to produce monoclonal antibodies from CHO cells. And you can develop up to 3 to 4 batches of monoclonal antibodies using C1 compared to CHO cells in the same amount of time. This slide is an example of the record levels of protein production that are possible from the C1 cells. The C1 cells have been demonstrated to produce very high levels of Fc fusion, monoclonal antibodies, antibody fragments, trispecifics, virus-like particles, antigens, and influenza hemagglutinin, which you see there on the left. This is just a sampling of the types and varieties of the potential proteins that can be produced at record levels faster and at lower cost than potential other cell lines. We have very focused business development efforts that are focused again on animal health, human health, infectious disease, and other market opportunities. Our business development strategy consists of external development programs that help advance C1 recombinant protein platform, which help fund our R&D initiatives and advance our science. This allows Dyadic to develop and advance our C1 recombinant protein technology at a low cost to the company. One of the models that we've demonstrated in our industrial business with licensing technologies, where we generated over $110 million between the license agreements with BASF, Abengoa, Codexis Shell, and the eventual $75 million sale of our industrial business to DuPont, is we can get funded proof of concept collaborations for specific therapeutic proteins that lead to potential upfront access fees, milestones, and royalty payments, or with small and medium biotechs, potential equity that leads to milestones and royalty payments, and then of course potential governmental and agency grants and contracts, which we've demonstrated through the IIBR and within ZAPI. And then of course we're working on internally Dyadic product development, where Dyadic funds high-value product candidate programs where C1 recombinant gene expression overcomes barriers of existing platforms with meaningful technological or commercial impacts. If you remember, that's the enablement story, where we can enable the expression of difficult to produce proteins that aren't moving into the clinic because of lack of productivity through expression, that we might be able to not only help move into the clinic, but move towards commercialization. An example is one of the products we're working on, we've talked about, is the glycoengineering of C1 cells, advancing nivolumab, the Opdivo biosimilar monoclonal antibody, as a proof of concept for the production of monoclonal antibodies. And of course, as we mentioned, we've deleted proteases in the engineering of knocking them out to reduce their background activity, to get greater stability and greater productivity. We've generated our own SARS-CoV-2 vaccine candidates and are now embarking upon SARS-CoV-2 variant candidates. We're working with the Oslo University on influenza, and we're in discussions with other infectious disease, and we've generated the initial cell lines for potential metabolites. Just to recap, we have a solid financial position, we have top-tier global partners, we have wholly owned programs, and we have a versatile, robust platform that can be applied to animal, human health, infectious disease, and other market opportunities. We have a strong competitive advantage, global business development efforts that's growing and expanding on a global basis. We have strategic partners with top-tier animal and human health companies, governmental agencies, and we have a growing experienced group of energized professionals, both as a management team and world-class board of directors and advisors. As mentioned previously, our management and directors have a very strong record of success and value creation. For example, Patrick Lucy was a founder and chief business officer of Phoenix Inc., a biotech company that was established based on the Phoenix expression technology, a microbial-based protein production platform. Phoenix was an NYSE biotech company until October of 2020, when the company was acquired by Ligand Pharmaceuticals for up to $516 million. Aaron Bose spent 34 years at Pfizer in the biomanufacturing of antibodies and other therapeutics. Barry Buckland spent 29 years at Merck in the vaccine space and the biomanufacturing. Our chairman of the board spent over 3 decades at Pfizer and was former chairman of Carex and on the board of directors. And of course, Ping Rawson, our CFO, has a very strong finance background. Ronan Chelit, our CSO, comes from Teva, Codexis, and has been experienced in working with C1 for over a decade. And then Matthew Jones comes from Lonza and GSK. And of course, myself in creating the value and building the company from the ground up. So this is a very focused management team that is highly energized to bring value to shareholders and to bring value to humankind on a global basis. There's a growing realization among scientists, business executives in the pharmaceutical industry, and governmental agencies that there's a global need for a more rapid, safe, scalable vaccine antigen and antibody manufacturing capability. The SARS-CoV-2 pandemic highlighted this unmet need. C1 cells can be used to develop affordable, scalable, safe and protective vaccines, antibodies, and other therapeutic recombinant proteins. The world is in need of better solutions to make healthcare more accessible and affordable to patients on a global basis. We believe that our C1 technology can help in filling that void. As shown here, we were successful on a global basis of getting C1 cells embedded into 9 programs for SARS-CoV-2 vaccines and antibodies. In addition, we're in discussions now with a variety of different companies that are interested in potentially using C1 to develop next-generation antibodies and vaccines. As mentioned earlier, we at Dyadic ourselves are working on and developing a potential tetravalent vaccine and potential embedded into programs for monoclonal antibodies, including potentially monoclonal antibody cocktails using C1 cells. Our vision at Dyadic is to accelerate the adoption and use of C1 cells to speed the development, lower the cost, improve the performance, while providing the necessary quantities of vaccines and drugs to a global population more affordably. Our strategy is to deliver C1 cells that produce recombinant protein antigen vaccines safely, effectively, and affordably to billions of people globally. To expand the infectious disease franchise to include bacterial, fungal, and parasitic pathogens. To advance immunobiologic vaccines and monoclonal antibody candidates into human clinical trials. To extend C1 recombinant glycoprotein manufacturing technology into new disease areas, including autoimmunity, degeneration, inflammation, metabolism, and oncology. To expand our global footprint with product capabilities for small and large-scale manufacturing footprints in North and South America, Europe, Africa, Asia, and India. To continue to hire experienced, intelligent, creative, ethical professionals, and to build our reputation as a potential enabler of global manufacturing, business development, strategic partnering, and commercialization of Dyadic franchise products, including potential Dyadic spin-out companies based on the platform technology that has many shots on goals in animal health, human health, infectious disease, and a growing number of other applications. Thank you for taking the time to listen to my presentation. Together we can make significant inroads in making healthcare more accessible and affordable to patients globally, while at the same time creating significant value for Dyadic and its shareholders.